Hello everyone, this is Dr. Pradibha from Department of Biotechnology, Kamaraj College of Engineering and Technology, and today we will be dealing with one of the very important molecules in biology, the antibody. We will be looking into the structure function relationship of antibody. We will deal in detail about the structural features that enable antibody to function the way it does. So, we will be going into what are antibodies. Let us revisit the immune system. The immune system offers a mechanism by which the body can combat foreign particles or foreign invasion, whatever it is. Basically, the vertebrate immune system does this beautifully, having these three properties. Number 1 is specific recognition of the foreign molecule. Number 2 is destruction of the foreign molecule or the pathogen. Number 3 is to develop a memory mechanism based on the encounter that it had accounted for. So, these are the three features that make immune system a more versatile one. And if you see, all of these functions of the immune system is mainly because of one group of molecules called antibodies. These molecules are also called immunoglobulins, and they are proteins secreted by the B cells that recognize specific regions on the antigens called antigenic determinants. So, antigenic determinants are actually parts of the antigen, not the antibody. They are parts of the antigen which the antibody is going to recognize. As you can see the picture on the right, when an antigen is being challenged to a group of B cells, and as you know, each B cell on its surface has a surface receptor which is capable of binding to a different antigen. There are around 50 million B cells that a vertebrate can generate in a single day, and each of them can bind to a different antibody or different antigen. So, their specificity varies vastly, and thereby whenever an antigen is challenged to the body, any of these B cell types will be the best fit for it. So, whichever has the best fit in recognition, the particular clone alone proliferates and it starts secreting more of the type of the receptor. So, basically B cell receptor is the immunoglobulin. When secreted, you call it the antibody. So, going into the structure of immunoglobulin, this is very common. The antibody is made up of protein. So, there are 4 polypeptide chains, and each of these chains are actually divided into different domains, which we will be seeing in a few slides later. So, these polypeptide chains, as you know, are the heavy chains and the light chains. There are 2 heavy chains and 2 light chains. The heavy chains can be of different types: the gamma type, alpha type, mu type, delta type and the epsilon type. And the light chains are called the L chains, and there are 2 identical ones in an antibody, and you call them the kappa or the lambda type. Functionally, there is no significant difference between kappa and lambda, but functionally there is a significant difference with these 5 different types of immunoglobulin heavy chains. So, they are connected by disulfide bonds which place them together, holding the molecule as a single entity. As I told you, these are the different subtypes of antibody: IgG, D, E, M and A. A is a dimer, M is a pentamer, IgG, D and E are monomers. Each of them have different effector functions. Looking into the domain structure of IgG, as I told you, the polypeptide chain of immunoglobulin is folded into multiple domains, and each of the domain, as you can see here, there are constant domains and variable domains. So, constant domains are the regions which do not basically interact with the antibody and antigen, and with the variable region is the one which actually interacts with the foreign antigen. The constant domains have similarity in sequences in primary structure. So, when you analyze different types of antibodies in their primary sequence, we identified that the sequence is highly conserved. So, variable domains are variable with respect to the sequence structure, but they are not uniformly variable, which means even within the variable domain regions, there are some regions which are conserved, which are constant, and there are some regions which are very variable in nature. So, we will be dealing with that and how it relates to the function of the antibody. So, these regions of greater variability are called the hypervariable regions, also called the complementarity determining regions. They are labeled as CDRs. There are 3 CDRs in a antigen binding site of each antibody molecule. So, you can find 3 in each site. They are called CDR 1, 2 and 3. As you can see, this is a detailed structure of antibody molecule. You can see the heavy chain constant region. This is the constant region of the light chain, constant heavy chain. So, as you can see, there are 8 constant domains in an antibody molecules, and you can see 4 variable domains in antibody molecule, and within the variable domains you can see hypervariable regions or regions of excessive amino acid mutations, where you can see hypervariability in their amino acid sequence. So, they are hypervariable regions. Antibody molecule is actually Y shaped when we draw it in 2-dimensional picture, but in reality the antibody molecule is connected in a hinge and it is flexible, okay, flexible for antigen binding. It is not a very rigid Y shaped structure, and you can see the domain structures here. Antibody molecules the domains of the antibody molecule interact with each other, forming a complex structure which is very important for its function. So, today we will be dealing about the secondary structural elements present in the antibody molecule. As we all know, protein has 2 secondary structural elements, one is the alpha helix and the second one is the beta sheet. Antibodies do not have alpha helices, they are entirely made up of beta sheet. So, it is a beta protein, beta strand comprising protein. So, as you can see, now we will see the structure of the immunoglobulin, basically the constant domain. So, in the constant domain, how is the structure with respect to the secondary structural elements? The constant domain is built from 7 different beta sheets. This is the topology of the beta sheet. You can see the N terminus here, and the direction of the beta sheet is shown in this topology diagram. There are 7 strands which pack into a barrel like structure to form antiparallel sheets that are tightly packed to call the immunoglobulin fold. So, as you can see, 2 strands of 4 and 3, they are packed against one another, thus forming a domain structure. So, this is how constant domain of immunoglobulin is formed. The loops that connect these structures are very short, sometimes like turns, okay, and throughout the framework regions are common throughout the constant domains. And this is the structure of a variable domain. So, variable domain is the one which is actually interacting with the antigen, that is the pathogen basically. You can see the variable region also has a similar 2 standard beta 2 beta sheets. In one you have 4 strands, and the other, instead of 3 strands in the constant domain, you have 5 strands in the variable domain. So, it is actually 4 plus 5 9 stranded beta sheet. These loop regions that connect these strands are called the hypervariable regions. It is actually these loop regions which interact with the antigen. It is not the entire variable domain that is interacting with the antigen. It is only the loop region that is going to recognize specific amino acids present in the pathogen or specific patterns in the pathogen or the antigen. So, CDR is a crossover connection from strand 4 and strand to strand 5. So, you can see CDR is in the same sheet. CDR 3 is a crossover connection between the green sheet and the blue sheet. So, as I told you, this is the domain structure, but antibody is not individual domain. Antibody functions when these domains are interacting. You can see constant domain of light region, light chain, and constant domain of heavy chain interacting to form an antigen binding site, and variable domain of the light chain and variable domain of the heavy chain, they have to interact, and all of this together form the antigen binding site. So, now we will see how these domain interactions are taking place. First, we will look into constant domain interaction. So, interaction between the constant domain. How do the constant domain of a variable chain interact with the constant domain of a how do the constant domain interact how do the constant domain of the heavy chain interact with the constant domain of the light chain. So, here we can see they interact by hydrophobic packing. The 4-stranded beta sheet of both constant domains, they pack against each other by hydrophobic interactions. You can see 4-stranded beta sheet of one domain interacts with the 4-stranded beta sheet of another domain. They form hydrophobic interactions and they form a packing. So, this is one domain, this is the other one, they pack together forming a stable interaction. So, this is how constant domain interacts. And now we will see how the variable domains interact. This is very important because variable region is the one which is going to bind the antigen. So, as expected, the 5-stranded beta sheet can interact with one another, or the 3-stranded beta sheet can interact with one another. There are two possibilities. So, actually what is in nature is, it is the 5-stranded beta sheet that is interacting with one another, they are packing. It is not the 4-stranded beta sheet. Why? Because in the 4-stranded beta sheet, if this were the case, the complementarity determining loops, they fall out of place and they are not together so that they can bind a antigen. Whereas, if the 5-stranded beta sheets are interacting with one another, you find that the loops are put together and actually they form a barrel like structure. They do not pack like the one we saw in constant domain. In constant domain, they pack against each other, whereas in the variable domain they form a barrel. So, the first strand is interacting with the 8th strand. So, the 10th strand, basically 8 strands they form a barrel. So, in this barrel, if you see on top of the barrel, all the complementarity determining regions or the CDR regions are aligned in such a way that an antigen binding site is beautifully formed. So, instead of packing, they form a barrel. So, it is this barrel which is actually recognizing the antigen. This is the front view. You can see the region on the topmost of the antibody is the one that is recognizing the antigen. So, from this we understand that the structural features built inside the antibody. The very important feature is that the amino acid sequences of the constant domains of immunoglobulins are highly conserved. They are called the framework regions and they rarely get mutated, because any mutation in the framework region will cost the structure of the entire antibody. So, when the structure is lost, probably its function will also be lost. So, these regions are highly conserved. In fact, the framework regions of the variable domains are also highly conserved. Only the mutation or the hypermutations are occurring in the loop regions, and that is why you can see so many different types of antibody molecules that can have great specificity for different antigens. So, that is in conclusion for this class. The structural features and arrangement of antibody enables its function of antigen binding. Thank you so much.